Signal transduction pathways through small monomeric G proteins. Small monomeric G proteins are crucial players in a variety of cellular processes, acting as molecular switches that cycle between active, GTP-bound, and inactive, GDP-bound states. These proteins are involved in signal transduction pathways, regulating a wide range of cellular functions, including cell growth, differentiation, proliferation, and apoptosis. The role of small monomeric G proteins, GTP as activity. These proteins possess intrinsic GTP as activity, allowing them to hydrolyze GTP to GDP, leading to their inactivation. GTP as activating proteins, GAPs, accelerate the GTP as activity, promoting the inactive state. Guanine nucleotide exchange factors, GEFs, promote the exchange of GDP for GTP, activating the protein. Guanine nucleotide dissociation inhibitors, GDIs, inhibit the dissociation of GDP, maintaining the inactive state. Major families of small monomeric G proteins. Several families of small monomeric G proteins exist, each with distinct functions. RAS superfamily, involved in cell growth, differentiation, and survival. Key members include ROS, Rho, Rab, and RAN. RAS activates the MAP kinase pathway, leading to gene expression and cell proliferation. Rho regulates actin cytoskeleton dynamics, cell adhesion, and migration. Rab controls intracellular vesicle trafficking. RAN, essential for nuclear transport. Our family, involved in vesicle trafficking, particularly from the Golgi apparatus. Signal transduction pathways. Small monomeric G proteins participate in various signal transduction pathways. 1. RAS-MAPK pathway, activated by growth factors and other extracellular signals. RAS, upon activation, recruits and activates RAF kinase. RAF activates MEK, which in turn activates ERK. ERK phosphorylates various target proteins, leading to gene expression and cell proliferation. 2. Rho GTP as pathway, regulates actin cytoskeleton dynamics. Rho activation leads to the formation of stress fibers and focal adhesions. RAC activates the formation of lamellipodia, involved in cell migration. CDC42 promotes the formation of filopodia, important for cell-cell communication. 3. RAB GTP as pathway, regulates vesicle trafficking between different cellular compartments. RAB proteins interact with various effector proteins to control vesicle budding, movement, and fusion. 4. ARF GTP as pathway, involved in protein sorting and vesicle budding from the Golgi apparatus. ARF proteins recruit coat proteins to initiate vesicle formation, regulation of small monomeric G proteins. The activity of small monomeric G proteins is tightly regulated by GEFs, GEPs, and GDIs. These regulatory proteins ensure that the G proteins function correctly and respond appropriately to extracellular signals,